Yes, everyone. So, hi, this is Harish Basi, and today we will be going to start with some of the left out portion which we were not able to complete in the previous session. So, we are going to complete all those things today, and we are going to solve some of the questions related to linkage, related to recombination frequency, and all of this thing. Okay, clear? Ready, everyone? Okay, great. So, now see here. Basically, the which is linkage is of two types. One is what? One is incomplete linkage. Incomplete linkage. And the other one is the complete linkage. Okay. So if I talk about the incomplete linkage, so what happens in incomplete linkage, sir? See, in incomplete linkage, uh, recombinant are formed. Like new combination is formed in incomplete linkage. New combination of phenotypes can be formed. Yes or no? Yes, sir. Okay, can be formed. Just it was happening in the first cross. You remember this one? See here, I'll show you. I will definitely show you. 1 second. This one, in this case, we can see the recombination were formed like 1.3%. The recombinant were formed in the second cross. Also the recombinant is formed. Yes or no? The recombinant were 62.8%. Sorry, the recombinant were 37.2%. So in these cases, the uh, linkage is what? It's incomplete, because still new combinations of phenotype is formed. But here's a thing, like the parental combination should all will be always more than 50. Parental combination will be more than 50. Okay, will be more than 50. But in case of the recombinant, the percentage of recombinant will always be less than 50. Okay, percentage or recombinant will be less than 50. Clear? So this is a case of incomplete linkage. Yes or no? Okay, sir. And it is very common. It's really very common. Like you can see it everywhere almost. But sir, there is one thing. What about the complete linkage? What about the complete linkage? 1 second. Should I arrange this mic properly? Can you give me 1 second? Yes or no? Obviously. So just give me a second. I will arrange this mic properly. And tada, I have this mic. Chalo. So next case is the complete linkage. What happens in complete linkage, sir? See, complete linkage. In complete linkage, there will be no formation of recombinants. Recombinants will not be there. No recombinants. Only and only the parental phenotype is produced. See, no recombinants, only parental types are formed. Only parental types are formed. Okay. Next, what's next? It's very rare. It is very rare, because recombination takes place generally. If I talk about, so recombination generally takes place every time. But if it's not taking place, this case can be considered as the rarest of rare. Okay, chalo. Now let's directly jump into some of the questions so that you will be able to understand the concepts very well. Okay, this session will be kind of short as compared to the other sessions. Okay, so see, I have a question for you, and that question says a plant is heterozygous. A plant is heterozygous for three genes, for three genes, okay, and let's name it as A, B and C. Okay, sir. Now it also says that in these three genes, two genes, which is A and B, which is A and B, is completely linked. Okay, is completely linked, which means, sir, what? Like whatever the gametes they are going to form, A and B will not segregate. Okay, then they are asking how many types of gametes, is formed. So now see, we have learned the Falkline method, yes or no? So we are going to follow that only, nothing new, just we have to take some precautions here. So if they are talking about the heterozygous condition for the three genes, so this will be the heterozygous condition: capital A, small a, capital B, small b, and capital C, small c. Okay, great. Yes, sir. This is the case only. Next, what, sir? Next, here we are going to use the Falkline method. Now, just remember that you have to consider the completely linked genes just as homozygous condition. Okay, so what is going to happen? There will be capital A, capital B, and small a, small b. This condition will be there. Don't segregate don't segregate A and B, because what, they are completely linked with each other. Okay, if they're going to make gametes, A and B is going to be uh, not separated. They will go inside the gametes as it is, like A and B will not be separated at all. So we have written A and B like this and A and B like this. Okay, great, sir. Next, what? Next we have capital C and small c, capital C and small c. Great. And then we will be having the gametes as A, B, C, A, B, small c, or what we can have, A, B, C, and sorry, A, B, capital C, A, B, capital C, and A, B, small c. Clear to everyone? Let me know if you have any doubts. What happened to my ears? Let me know if you have any doubts anywhere. Is this question clear? What we have done, sir? We haven't separated this A and B. Why? Because they were linked with each other. They are completely linked with each other. Okay? Great, sir. Now, let's move on and let us try some other questions as well. So, let's have the other question. The other question says, should I give this question little hard? Okay, okay, chalo. See, in a plant, it says in a plant, 20% recombination is formed. Okay, how many percentage is recombination is there? 20%. 20% recombination. 20% recombination is found or formed. Okay, great, sir. Between which genes? Between genes of small a and small b. Okay, take it. Then I have some of the options. Then let's see. Number one is the option says, number one option is plus plus and AB. So it say 80% of plus plus will be formed, like A plus B plus, or 20% recombination will be formed. Next we have plus plus 40%, this will be formed, and then AB will be formed as like this. See, One second. Plus A 10% and plus B 10%, and then we have AB as 40%. What is this plus, sir? It's nothing. It's nothing. It's just the way of writing A plus and B plus. Okay, don't get confused. Third option says that plus 20% and AB will be 80%. And fourth option says plus. Let's leave the fourth option. Let's have these three options only. Okay, now let's solve the question. Did you understood the question? What is the question is saying that in a plant 20% recombination is formed between genes A and B. Then which one is correct? Is A plus B plus, which is going to be formed, will be 80%, or AB will be 20%, or plus will be 40%, or plus A will be 10%, plus B will be 10%, right? In the question it's also given like the parents are kind of pure. Okay, the parents are kind of pure. So let's see here. Let's see. In the question it's given the parents are pure. So let's take, sorry, parents are heterozygous. I'm so sorry. The parents is heterozygous. Is heterozygous. Okay. If the parent is heterozygous, then how we are going to form? See, if the parent is heterozygous, that means plus A plus B is there, and if they're going to make the gametes, then what type of gametes we will be forming? We will be forming the gametes as plus, then we will be having plus A, yes or no? Then what we can have? Plus B, and then what we can have? A and B. Okay, how we have formed? Nothing. See, plus A and plus B we have written. Okay, so plus can be plus. Okay, this and this can be plus and A, right? And again this plus and this plus can give us plus. Acha, we have done that. One second. Okay, then what's this plus can be formed with plus B, and this A and B can be formed with AB. So this is how we have made the gametes, right? Okay, great, sir. Now it's saying that 20% of recombination is formed. 20% of recombination is formed. So what is recombination, sir? What is recombination here? What kind of recombination you can observe? See, earlier if I talk about the parents, okay, earlier if I talk about this particular parents, like how this parent was formed. So let's get back into little earlier. So the parents would be what? Plus and plus crossed with AA and BB, and when they are crossed they are giving what? They are giving plus A and plus B, right? And when it is making the gametes, when this particular thing is making the gametes, then what we are getting? This thing. Is there any new combination which is formed? Yes, sir. This is the new combination. This is the new combination, right? Clear? Okay. So new combination with is what? Recombination. So total percentage of recombination was 20%. Total percentage of recombination was 20%. Yes or no? So for individual it will have 10%. It will have 10%, right? And this will have 40%, and this will have 40%. Okay, so the answer to this question will be what? We will be having this one, because plus is 40, AB is 40. Why this is 40? Because this were the parental combination, right? And this is the combination which is newly formed. So this will be of total 20%. Okay, understood? Understood this question? Great. Now let's move on and let us talk about the another question. Now the question says that a plant has heterozygous heterozygous genotype and 10%. If you didn't understood that question, this question, from this question your concept will be very strong. So a plant has heterozygous genotype and 10% recombination is possible, and 10% recombination is possible between A and B. Okay, and in total 800 gametes is formed. In total 800 gametes are formed are formed in which the number of gametes, sorry, in which the number of, not gametes, let me write here, in which the number of A and capital B types of gametes must be. Types of gametes must be C. So here basically what it's asking, it's saying that 10% is recombination is happening. So what will be the percentage of A and capital B types of gametes? Okay, see here. So it said that the plant is heterozygous. That means what? Capital E, small a, capital B, small b. Okay, sir, great. Now what will be the gametes? Let's make the gametes here. How many types of gametes will be formed? See, if we use the fork line method here, so how many types? Capital A, let's bifurcate it, right? Let's bifurcate it, capital A and small a. See, in this question they haven't said that complete linkage. How many recombination is forming? 10% of recombination is forming. So we can make the fork line method and we can bifurcate it. If they would have said AB is completely linked, so rather than bifurcating A and A like this, I would just have written AB and AB, right? But in this, recombination is happening, so we can use the normal fork line. Here we have B and here we have small b. Again, here we have B and here we have small b. So the gametes which we are getting is AB, is A small b, is small a capital B, is small a small b. Okay, great, sir. Now it's saying that 10% of recombination is happening. So what is recombination here, sir? What is recombination here? Yes, the major question is which one is the recombination here, whether AB is the recombination or AB, this AB, or is this the recombination, or this AB and AB is the recombination. So here if I talk about the recombination, so we have the recombination as small a, capital A, small b, and small a and capital B. Yes or no? Okay. They both have what? They both are of 10% each. Sorry, 10%. So total 5% and 5% we are having the recombination in total. And then how much we are getting? And the question is saying that what type of, find the number of gametes which is having this much recombination, right? See, a plant has heterozygous genotype and 10% recombination is possible between A and B. Total gametes 800. So what we have to find out? Number of A and B types of gametes must be. See here. So 5% recombination is happening in A and B. So 5% upon 100 into total number of gametes which is being formed, and it will be what? Tuck, 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 8 5s are 40. So 40 gametes will be what? It will be recombinant, having the genotype as small a and capital B. Clear to everyone? Any doubts anywhere? Let me know in the comment section. Any doubts? Any doubts, guys? Any doubts? No doubts? Great. If you don't have any doubts, that's very, very good. That is really very good. Okay, now let's have a few more questions. Let's have few more questions. See, the next topic, before that, let's have a next topic, and after that, let's solve some questions, because to solve some questions, you need the formulas also. Okay, so we have a topic, a very small topic, which says recombination. Recombination frequency. Recombination frequency. Now, what is this recombination frequency? How we can calculate this? So, this recombination frequency is nothing but can be calculated as number of recombinants divided by parental type plus recombinant, which is total number of gametes or total number of outcomes, plus recombinant multiplied by 100. Okay, great. Now there is one more thing. Recombination frequency is what? It is measured in the form of 1 cm, 1 centimorgan. What is this 1 centimorgan? See here. If I talk about the 0.01 or 1% recombination 1% recombination frequency is considered as 1 map unit or 1 cm. That means if there is 1% of recombination frequency, we are going to call it as 1 cm. Okay, clear? Right, sir. Now let's, recombination frequency can be equal to 50 and less than 50. It can't be more than 50. Okay, so I should write here like this. It can't be more than 50%. Okay, great. Now let's get back to solve some of the questions. Like more 10 minutes and this lecture will also be over, and then we will be left with sex determination and sexual sex inherited characters. Okay, so that we will do in live class. So tomorrow you will be completing the chapter reproductive health. There are 2 sessions of those chapters. You have to complete it, because as soon as I will be starting taking live classes, I'll ask the questions in between of your classes. Okay, so see, a plant is heterozygous. A question says a plant is heterozygous for genes A and B, and it produces 1200 pollen grains, 1200 pollen grains, out of which 500 are of capital A, capital B type. What must be the distance between 2 genes? Okay, let's solve this question. See, so what's happening is that it's saying that total 1200 pollen grains are formed. Okay, out of which 500 are capital A and capital B kind of thing. Okay, great. Now what must be the distance between these 2 genes? Clear? Okay, sir. So see here, if I am talking about the parent, so parent is this, right? Heterozygous hai for A and B, so parent will be this, and this is producing total 1200 chromosomes, and out of 500 they are having capital A and capital B, capital B. One second, I should write here this one now. One second. Capital A and capital B types. Okay, clear. Now check. So let's make the pollen grains. If we are making the pollen grains, that means we are making the gametes. So here will be capital A, one gamete. Okay, and sorry, capital A, small capital A and capital B, one gamete. Then we will be having capital A, small b. Then we can have small a, capital B and small a, small b. How, sir? Again with the fork method. Okay, and now it is saying that this one is of 500. That means parental type, original type is of 500, and this combination is again of what? It is also of 500. And recombination will be what? 200 each, sorry, 200 in total, because total pollen grain is what? 1200. So 200 pollen grains in total will be having recombinant one. Total will be 200. So this one will be what? 100, and this one will be what? 100. Okay, so what they were asking? Distance between two genes. Okay, distance between two genes. So how to find the distance between two genes? With the help of this formula. So number of recombinant is what? Number of recombinant is 200. Total number of progenies, recombinant and parental type 1200, multiplied by what? 100. So when you are going to solve this, so it will be 200 divided by 12, and when you are going to solve this, so 2 6 are 12, 2 6 are 12, 50, and then you're going to get 16. Basically 50 by 3 you're going to get here. If you also 50 by 3 you're going to get 112. Yeah, yes, 50 by 3 you're going to get, and 50 by 3 will give you the 16.6%. Okay, clear to everyone? And if I am talking about 16.6 percentage, which means what? Which means that 16.6 centimorgan, right? Okay, now let's have one more question for this. Just another question. Distance between A and B is 30 centimorgan. Okay, C should be small. Over a chromosome, there is one chromosome, and in that chromosome the distance is 30 centimorgan. Okay, over a chromosome which is heterozygous, for A and B, and produces what? Produces 600 gametes. 600 gametes. Then what you have to say? How many? What you have to find? How many of them must be AB and AB types. Okay, see. So this earlier we were given the gametes, and we have been asked about the distance. Now we have been given the distance, and we have to ask tell about the gametes. Okay, so see, again, that is heterozygous. So let's have capital A small a, capital B small b only, right? It's saying na, this A and B is 30 morgan, heterozygous hai, and they produce 600 gametes. How many of them will be this and this, right? See, so let's make the gametes. Capital A, capital A, small b, small a, capital B, small a, small b. Okay, now if the distance is 30 centimorgan, which means what, sir? Distance between A and B is 30 centimeter, right? That means recombination frequency is what? 30%, yes or no? See, it's saying that recombination this 30 centimeter, centimorgan represents what? Recombination frequency, which means 30% recombination is happening. Okay, 30% is recombination is happening. So both for single, this is having 15%. Are, why didn't the color changed? This is for single 15% and this is for again 15%. Okay, and this will become 35% and this will become 35%. So we have been asked about what? We have been asked about this and this. So A, what? One second. A and B. Okay, why I have written capital A here? A and B. So let's calculate 35 divided by total number of what? Total number of gametes, what? 600 multiplied by 100. So it will give us what? It will give us around 635 upon 6. Approximately it will give us 210 gametes. Okay, pollen grains. And then the question asked about AB types. So AB types, this is the AB types. So percentage is 15. 15 divided by 600 into 100. And then when you're going to solve this, you're going to get approx 90. Clear? What? What I have done? Sorry guys. Sorry. I did a blunder mistake. Oh my god. One second. Bachcho, 1 second. Sorry. 600 multiplied by 15 upon 100. Okay, this is going to give us 15 6 the 6 5 the 30, yeah, 90. Okay, for AB. And then we are going to have capital A and capital B. I'm so sorry. I am so sorry. So capital A and capital B again, 600 multiplied by this will be 35 upon 100. Okay, so this we are going to get 210. Yes or no? See, check this one now. Is it clear? Is it clear now, people, students? Is it clear now? Okay, so 90 of the gametes will be containing capital A and small b, and 210 types of gametes will be having capital A and capital B. In this way, we can solve the questions regarding the recombination frequency. Okay. So I hope that you enjoyed this session, and in case of any doubts and any questions, let me know in the comment section. Okay. I understand that these are the recorded sessions which in which I may not interact with you as I am interacting in the live classes, but instead of cancelling the classes, I prefer to record the sessions so that your flow shouldn't be hampered, my flow shouldn't be hampered. Okay, and when I will be back on 26th, we will again start the live classes. And in case you have any kinds of doubt in any of these videos, in that class, whatever topic we will be learning, I'll take your doubts first and we'll try to solve those doubts. Okay, so don't worry, you have watched this lecture, and only 2 sessions of genetics is left, which is sex determination and the inheritance of sexual characters. I really want those topics to be done online so that I can explain lots and lots of things to you. So we will do these sessions on one session we will do on 26th and one session we will do on 27th. But before that, you need to complete the reproductive health. I won't give you 2-day holiday to enjoy. Okay, so I have shot the reproductive health video also. Complete that. That's a small chapter. Let's do it in 2 days only. Okay, chalo. Bye bye everyone. See you in the next class of reproductive health.